Good day everyone, I'm Joyce Joy G. Agnes and today I'll be presenting the study entitled Ocular Stem Cell. This is the abstract of the study. For the introduction, stem cells are capable of replacing damaged or diseased cells under certain circumstances. Regenerative medicine or stem cell-based therapy has reached a state where ocular tissues damaged or disease or injury can be repaired or regenerated. The ease of access for the therapeutic procedure as well as follow-up together with its immune privilege status makes the eye an ideal organ for studying regenerative medicine. Such as therapy involves various methods where stem cells are injected into cellular and extracellular matrix microenvironments. Corneal epithelial cell transplantation has been the most widely used stem cell-based therapy, followed by bone marrow transplantation. Stem cell-based treatment in ophthalmology follows either a cell replacement therapy strategy or a strategy involving trophic factor-based guidance cues. Throughout treatment, outcome depend on our depth knowledge of the disease, the source of stem cells, the mode of treatment, and the plausible mechanism driving the therapeutic outcome. In this review, they discuss region-specific stem cell populations and their respective function in cell-based therapy. They also address possible hurdles to therapy and the means to overcome these in our pursuit of regenerative medicine application in the field of ophthalmology. This picture shows the cornea, limbus, and stroma. The cornea is at the outermost surface of the eye and safeguards transparency, which is crucial for vision. For the therapeutic implications of cornea, LESC deficiency is pathological, either partially or completely, and is caused by either mechanical injury or chemical and thermal burns or acquired by diseases such as aniridia and Stevens-Johnson syndrome. Treatment of such condition involves LESC transplantation therapy. Among non-limbal cell types, cultured oral mucosal cells and conjunctival epithelial cells have been transplanted to treat limbal stem cell deficiency in humans. This is the conjunctiva. The conjunctiva, apart from being a barrier of pathogenic entry, is a highly vascularized connective tissue that provides channels for the proper flow of nutrients and fluids. Conjunctival cells undergo renewal like a corneal epithelium, but the source of the stem cells for this remains elusive. Therapeutic implications of conjunctiva. Conjunctival autografts, oral mucous membrane grafts, nasal turbinate mucosa grafts, and amniotic membranes are often used to treat conjunctival stem cells deficiency and scarring. Conjunctival cells cultured and amniotic membrane have been used for cell transplantation in patients with limbal stem cells deficiency. This picture shows the iris. The iris divides the space between the cornea and lens into anterior and posterior halves. The stroma and the vasculature of the iris are developed from the anterior region of the optic cup. The therapeutic implications of iris, though the iris pigment epithelial cells have the potential to be used in cell-based therapy, not much work on validation and quality assessment has been done. This is the ciliary body. The ciliary body produces the aqueous humor and involved in regulating the aqueous flow, blood flow, intraocular pressure, and maintenance of the immune privilege status of the anterior chamber. Therapeutic implications of ciliary body. Bayes and colleagues showed that clonally derived retinal stem cell progeny from ciliary epithelium can differentiate into mature rhodopsin positive cell using a combination of exogenous culture additives. Inouye and colleagues demonstrated that modulation of the retinal transcriptional factors OTX2, CRX, and CHX10 increases the potential of retinal stem cell progeny derived from the ciliary margin of the adult human eye. This is the trabecular meshwork. The trabecular meshwork, or TM, is a tissue between the cornea and iris in the anterior region that is responsible for the drainage of aqueous fluid. The balance between aqueous secretion and outflow determines intraocular pressure, which is a risk factor for the development of glaucoma. TM cells help to remove debris in the circulating aqueous humor. The therapeutic implications of trabecular meshwork is that lowering the intraocular pressure is an aim of treatments for glaucoma. A very recent study reported that stem cells isolated from human TM and expanded in vitro showed evidence of the ability to home to mouse TM and differentiate into TM cells in vivo. The expanded TM stem cells express the stem cell markers ABCG2, Notch1, and MUC1 and were positive for expression of the TM marker protein CH13L1. These TM cells were multiponent and had phagocytic properties. Some groups are working on transplanting TM cells or TM progenitor cells combined with an argon laser trabeculoplasty as a novel cell-based therapy for glaucoma. This is the lens. The lens is composed of the lens capsule, epithelium, and fibers, and like the cornea, is transparent. Lens stem cells are hypothesized to reside in the lens capsule, although they have not yet been identified. It is plausible that they come from the ciliary body, which is anatomically close to the lens. Therapeutic implications of lens. Lens progenitor cells often derive from human ESCs or embryonic stem cells, as well as induced pluripotent stem cells. Lens stem cells are presumed to have a role in maintaining lens transparency and might be important in cataractogenesis or other lens abnormalities. This image shows the retina. The retina represents the connecting link between visual input and image processing in the brain. Retinal diseases mostly result in reversible damage to the visual pathway. Several studies in animal models have achieved some amount of success using the transplantation of photoreceptors, endothelial cells, and retinal pigment epithelium, or RPE. Therapeutic implications of retina. Diseases in the inner retina include retinopathy and optic neuropathy, which cause damage to the retinal ganglion cells and amacrine cells. Cell transplantation in a retinal degeneration model has shown promising visual outcomes, but the extent of the curative effect remained unclear. The injected stem cell integrated into a retinal and subretinal microenvironment modulated the differentiation of different cell types. These transplanted cells integrate in a temporal independent manner that occurs only during radiogenesis. Photoreceptors and retinal pigmented epithelium. The current stem cell-based therapies for retinal diseases focus on supplementing or replacing photoreceptors and RPE in the affected retina. Therapeutic implications of RPE. Several groups have shown the capacity of human ESCs to differentiate into RPE with variable success rates. Recently, the differentiation efficacy was increased 30-fold by adding vitamin B3 and activin A protein. Murine disease models such as liver's congenital amaurosis rat have been used to study transplantation with differentiated retinal precursors. No teratoma formation was observed, but the curative outcome needs to be followed up. This is a choroid. The choroid is derived from mesoderm and neuroectoderm. Choroidal stem cells obtained from urine studies reveal mesenchymal stem cells properties, expressing markers such as SCA1, CD90.2, CD44, CD105, CD73, ABCG2, 6-2, Notch1, and PAX6. Therapeutic implications of choroid. Choroidal and scleral cells have been differentiated into retinal lineage cells under laboratory conditions. Further studies are needed to understand the biology as well as other attributes such as differentiation and proliferative aspects of these cells before they can be used clinically. This image shows the sclera. The sclera is continuous with the cornea and is composed of fibrous material with viscoelastic properties. It is responsible for maintaining ocular pressure. Scleral stem cells have a mesenchymal origin and express ABCG2, 6-2, PAX6, and Notch1. Therapeutic implications of sclera. Diseases that cause distension of the sclera, such as myopia, might be repaired using sclera stem cells. This might provide a source of bioengineering sclera for cell-based therapy, but more studies are needed to determine the role of scleral stem cells and treatment implications. This image shows the orbit. The orbit provides a scaffold for the eye. It is a bony cavity that contains the eye, optic nerve, extraocular muscles, nerves, fat, and lacrimal gland. In the recent years, adipose tissue stem cells have been studied extensively, and some research has been done on orbital adipose stem cells. Diseases such as thyroid eye disease and aggressive malignant tumors lead to neurological consequence and blindness. Therapeutic implications of orbit. Destruction of corneal epithelial cells results in loss of vision. Stem cells isolated and expanded from the limbal area of the ocular surface are able to repair the corneal epithelium. However, obtaining healthy limbal stem cells and immune tolerance are still issues. Recently, Lenin colleges used orbital fat-derived stem cells to promote corneal tissue regeneration through a non-surgical route. Topical administration of fat-derived stem cells resulted in the inhibition of inflammation and corneal revitalization. Let's go for the conclusion. They provide comprehensive detail on the localization of ocular stem cells and explain the therapeutic potential of each. Ocular diseases can be classified into vascular defects, anatomical defects, and neurodegenerative defects. In order to address these defects, regenerative medicine using cell replacement strategies could be highly beneficial and effective. Identification of the proper sources of stem cells is the first step toward this, followed by the isolation and characterization. Ophthalmology is the only branch of medical science that has so far gained from the field of regenerative medicine. Limbal stem cell transplantation is the only other cell-based transplantation procedure other than bone marrow transfusion that has been approved for the patient care. Table 1 ocular stem cell shows the location, functions, markers, and therapeutic development stage. Table 1 and 2 provide summaries of the present and future prospect of stem cells for ocular therapy. This is the continuation of Table 1. Table 2 for future prospects for stem cell use for ocular cell therapy. Figure 1 depicts the location of stem cells and their clinical application status. Figures 2 and 3 highlight stem cell sources used in ocular cell therapies for specific diseases. In order to harness the potential of stem cell-based therapy to provide and restore sight in blind patients, the safety of the cells needs to be studied in detail. For the successful utilization of stem cells for therapeutic purposes, small molecules can be incorporated with or conjugated into them before transplantation to promote specific differentiation pathways. This cell serves to replace damaged cells and produce cytokinesis growth factors and other trophic molecules. This shows the status of ocular and non-ocular stem cell transplantation for anterior surface disorders of the eye, MSC. This is the continuation of status of ocular and non-ocular stem cell transplantation. And for the abbreviations and for the reference. And the tense of my report entitled Ocular Stem Cells. Thank you for listening. Again, I'm Teresa Joy G. Agnes from Opto 1-1.